Okay, ladies and gentlemen, I think it's time to get started. Good morning. Welcome to you all, and thank you for being here today. Before introducing our speaker, just a few technical words for those of you who are online. As usual, you are in a webinar mode, which means that your mics and cameras are automatically switched off. If you have any technical issue, please use the chat to report and we will try to help you solve the problem. And if you have questions to our speaker, you can use the Q&A or Q&R tab to ask your questions, and we will allow you to switch on the mic to ask the question directly. So now it is my great honor and pleasure to introduce our distinguished speaker, Professor Jane Memmot, who has kindly accepted our invitation to give this year's Louis Thaler lecture. Jane Memmot is Professor of Ecology in the School of Biological Sciences at the University of Bristol. Her scientific career began in 1985 as a PhD student and tropical entomologist at the University of Leeds, and there she studied the community ecology of sandflies in the rainforests of Costa Rica. In particular, she investigated how sandfly species distribute depending on the interactions with trees, their host animals, other sandfly species, but also conspecifics through mating behavior. After obtaining her PhD in 1989, she held 2 postdoctoral positions at the Imperial College London in Silwood Park. And during these, she developed what would become the foundation of her scientific career, that is the first plant herbivore parasitoid food webs, focusing initially on leaf miners and their parasitoids, again in the rainforest of Costa Rica. She then moved on to biological control of non-native invasive species in the tropical dry forest, this time in New Zealand. After this, she was appointed as a lecturer at the University of Bristol in 1996 and became a professor in 2007. There she established a group working in the area of community ecology, using mostly ecological networks to study a wide range of ecological processes, and this across various ecosystems, from English meadows and Scottish islands to Hawaiian swamps, the mountain of Nepal, North African fynbos habitats, and urban areas. Her research focuses mostly on understanding ecological networks and how they respond to environmental changes, and this in various critical areas, including pollination ecology, agroecology, invasion ecology, or urban ecology. One of her many significant contributions is the publication of the first plant pollinator food web, showing community-level links between plants and their flower visitors for a nature reserve in Somerset in the UK. She is also a strong advocate for creating urban habitats that support pollinators by growing wildflowers to provide essential resources such as nectar and pollen. This not only supports biodiversity, but also helps to replenish nectar resources that have been depleted by agricultural practices. Other important contributions of Professor Memmot include the interaction of non-native species with plant pollinator and plant herbivore parasitoid networks, also in relation with the introduction of biocontrol agents. Last but not least, she proposed the concept of networks and constructed the first one by combining a range of different ecological network types, including pollinator, seed dispersal, host parasitoid networks, as well as trophic root webs. But do not think that Professor Memmot's commitment to science is limited to research. She has also contributed enormously to the profession. She was head of the School of Biological Sciences from 2012 to 2016, where she managed the move of the entire school to a new building, and has been director of teaching in the same school over the past 4 years. She has also served as president of the prestigious British Ecological Society, as an executive editor of the Journal of Applied Ecology, and as a member of the board of editors of the journal Science. Professor Memmot's contributions have been widely recognized. She has received several prestigious awards, including the President's Medal in 2011 and the Marsh Ecology Award in 2015 by the British Ecological Society, and the prize of the Royal Society for the Protection of Birds in 2017 for her publication in Nature, on the reconstruction of the dynamics of floral resources in Britain since the 1930s, using historical data on nectar productivity. In 2021, she was made an officer of the Order of the British Empire for her outstanding services to insect pollinators and ecology. And just 2 years after, she was elected as a Fellow of the Royal Society. What makes Professor Memmot's work particularly noteworthy is her dedication to translating research into practical applications. She collaborates closely with farmers, conservation practitioners, and policymakers to ensure that her findings lead to real-world solutions that benefits both nature and human well-being. We are really privileged to have her here today to share her insights and her expertise. So please join me in welcoming Professor Jane Memmot and enjoy the lecture.
Right, it's an absolute pleasure to be here today. So I'm delighted to be giving this talk. As Bruno says, I'm going to be talking about pollinator conservation and ecology. And I'll start with just an overview of where we're going to go over the next kind of 50 minutes or so. So I'll give a brief kind of resume with pictures this time of how I came to be working on pollinators. I'll impress upon you, just in case you don't know, why pollinators are so important and so wonderful. And then give an overview of the community ecology approach that we use for most of the projects that run in the group. And then the bulk of the talk will be on to the three research projects, three of my favorites. So there'll be urban pollinators, the urban pollinator project that ran for about 5-6 years. One on conservation at the landscape level, because most conservation projects these days, many of them look at landscape level processes. And then the project that's actually just started it's kind of been running on a shoestring the last few years, but we just got funding to run this project looking at the impact of pesticides on pollinator communities. And so I'm particularly excited about this one because it's the new project in the group at the moment, and will run the next 4 years. So there's no data yet, but there's lots of ideas and lots of things happening in the field. And then I'll wind up with a little bit on how to bring more pollinators into your lives. Pollinators are wonderful, they are good for you, they make you happy, you need more of them, okay? So we'll do that at the end. And then some brief conclusions. So starting off, how I came to work on pollinators. So this is a very young me, aged about 5. And as a little kid, I loved natural history. And I was pretty wide-ranging in my interests. But the things that really got me were insects. And to this day, I'm still mostly an entomologist, even though I work a lot on plants. And we include birds and mammals in some of our networks. But age 5, my first year at school, I found one of those caterpillars. We call them woolly bear caterpillars. I took it home and I put it in a jam jar and fed it, and this astonishing moth came out. And you know, to this day, that is still the most magic process ever. Beats anything on the internet available today. Okay, so if you've got kids, get them rearing caterpillars. It's truly addictive, and I am still rearing caterpillars to this day. So that's early on, very early on. PhD, as Bruno said, this is a phlebotomine sandfly, a wonderful little critter that does spread diseases, but I was working on the community ecology simply because it got me to the rainforest. I wanted to go to the tropics, as you know, I'd never flown until this point. So I flew out to Costa Rica and I worked for 3 years on these amazing phlebotomine sandflies, on their community ecology. Postdoc number 1, put together the first ever quantitative food web working with Charles Godfrey at Imperial College. I swapped a fancy American fuel station for a big tent under an even bigger tree. I've lived a pretty adventurous life for a few years. You got all my rearing pots in the front there, and I did live in that tent for 6 months a year. I'm still at Imperial College at this point, working on the second postdoc, working on the biological control of weeds, mostly in New Zealand and Australia, and a little bit of Kenya. And everything you can see in that photograph is a weed. All those yellow things are weeds, 2 species of European and American weeds. I then got a lectureship and started running my own group, and this is Laurie Henneman, who's one of the first very wonderful postdocs that have come to work with me. And I've been there ever since, working up to where I am today. At the time I arrived in Bristol, I actually, I'd been living a life as a very, I traveled all over the place. I was living out of a rucksack. I got a job and had kids, had a dog, all at the same time. Suddenly had to kind of reinvent myself. The kids are now, they graduated last year. They are somewhat bigger than that. The dog is sadly no longer with us. That's Ruby there. But at this point, I had to reinvent myself because I couldn't continue as a tropical person always living out of a rucksack. And I was bored of traveling at this point, so it suited me really well. And the field I moved into and where I still am today is kind of agroecology and pollination. And the sorts of questions I was able to ask were every bit as interesting and adventurous as those in the tropical rainforest. But it meant I could pop out for an afternoon to a local farm and still do research. So it was a happy transition. And to this day, I've worked mostly in the UK, but I always have one adventure, just one little adventurous project there. So over the years, I did a lot of work in Portugal and the Azores, simply because there was a stream of very delightful Portuguese PhD students that came to Bristol. And through them, I worked in these lovely places. And then the last kind of 5-6 years, we've been running a big project in Nepal, working with smallholder farmers, and also in South Africa, where the plants are just bigger and better than anything you've ever seen before down in the Cape on South Africa. So that's where I kind of come from. That's the kind of journey. So let's just have a very quick revision session on why pollinators are so important, and then go on to look at the community ecology approach that we use. So 87.5% of plant species worldwide need insect pollinators. Okay, they are really important if you're a plant. Pollinators are also essential for the production of 75% of crop species worldwide. So for humans, plants are really important. Our calories come from wind-pollinated plants like rice and wheat and maize and things, but the micronutrients, the important stuff in our diet, come from insect-pollinated plants. So pollinators are really important for producing those plants. And they're also highly charismatic creatures, which is actually just a bonus. So when you're dealing with the public and politicians, they like pollinators in a way that they didn't really like phlebotomine sandflies very much, or even parasitoids, which are wonderful as well. They don't quite get those. But I had a quick look on Google last week, and these are just some of the, those are all pollinators. They are truly awesome. And the insects even, the bumblebees and the ashy mining bees, they are truly beautiful things. And by the time you get to South Africa, and there's malachite sunbirds out there on the most ridiculous protea-type flowers, it's just mind-blowingly beautiful. Okay, so that's just a perk of working on pollinators. Many of them are very, very beautiful things. Now, last bit of the introduction. I want you to imagine you're sat in a meadow, a very simple meadow consisting of just 4 species of flowering plants. And I'm going to put those flowering plants just visually as 4 squares. The bigger the square, the more abundant the number of flowers. So we've got some common plants and some rare plants. And those plants are visited by 2 species of pollinator. Again, a common one and a rare one. And what we do is we map the interactions between all sorts of organisms, but in this case, plants and pollinators. So here we've got a pollinator visiting, a fairly specialist one, visiting just 1 species of plant versus a pollinator that's feeding on 3 species of plant. So that's a very simple cartoon of what the real world looks like. And even if you go to just, the simplest meadow, this little Somerset meadow in Bristol, this was the very first of these plant pollinator networks I put together. So that's from a very normal little nature reserve. And as you can see, it's more complicated than the little network, but it's just the same principle. It's just species are nodes, and they're linked together by these lines, and the thickness of the line tells you something about the frequency of interaction. And that basic kind of data, which is visualized there, but it just goes into a matrix or a list of connections, is kind of the everyday sort of data that we collect. Now, that Ecology Letters web is still pretty simple compared to what happens in the real world, because pollinators don't operate in isolation. They can be part of a much bigger network. So this is the Norwood network, which was this network of networks we did where we linked pollinator networks with herbivore networks, with parasitoid networks, with birds and mammals, and even the fleas and lice on the birds and mammals. One of my abiding memories of that project are the postdoc sat there combing the mice with a toothbrush to get the fleas off them. So we're looking at all sorts of different links in all sorts of different places. So you can try and capture, this is an organic farm network, and it was a way of just showing how biodiverse farmland can actually be, and what are the kind of, determinants of biodiversity in those habitats. And then pollinators are also food, as well as being, incredibly important for pollinators. They get eaten by all sorts of things. So this little network shows you, it's a heathland network. You've got plants at the bottom, and then all the bumblebee species, and then all the things that eat the bumblebees, which are a mixture. In this case, we've got parasitoids, pathogens, and parasites, so things living on the inside of the bee and the outside of the bee. And of course, things like birds and bats feed on those too. And in this talk, I'll start with the Urban project, which looks at just the simple plant pollinator networks. I'll then move on to the Landscape Net project, which looks at pollinators with herbivores and parasitoids. And then we'll end with something which is looking at cascading effects through the pollinator food chain. So if you improve your pollinators, you also could end up with more bats and more canopied parasitoids or something. So that's where we're going. So let me start with the first of these three projects. And actually the bulk of the talk's on the first two because they're mostly finished. So the Urban Pollinator project. So I grew up in a variety of urban habitats. And as a kid, there's a lot of biodiversity in urban habitats. And they're not tropical rainforests, but there's still a lot you can do as an ecologist. And because most people live in urban habitats, it's the place that most people are going to see biodiversity. And they can be surprisingly good for biodiversity as well. So these are the questions we're asking on this project. So there are three questions. We wanted to know where exactly the pollinator biodiversity was in the UK. Is it in urban habitats, farmlands, or nature reserves? Because although there was a fair bit of data on those separately, nobody had systematically compared them to ask, well, where, are pollinators in the countryside, are they in towns, and in nature reserves? And then beaming in on cities, we wanted to ask, well, where are the best places in cities? Because cities are really variable habitats. So we wanted to know where the hotspots for pollinators were. And then finally, we wanted to know what we could do to improve pollinator biodiversity and abundance in urban habitats. Okay, how do you make cities better for pollinators? So this is the team. This was it was a consortium project, so I led the consortium from Bristol, but hugely aided and abetted by three other academics. So Simon Potts in Reading, Bill Coonan in Leeds, and Graham Stone in Edinburgh. We worked really closely with taxonomists because pollinators, even in Britain, which is pretty nature depleted these days, there's still huge numbers of species, and you need professional taxonomists to do those solitary bees and lots of the flies. And then what actually made this project particularly fun was we worked really closely with the conservation practitioners in each city, because that teamwork, that synergy between academics and practitioners is a really powerful one, because academics have the ability to measure things and test things and gather data very systematically. But conservation practitioners own and run nature reserves, so they have the ability to change what happens in the field. So working with them is both really fun and an incredibly useful way to work, with advantages to both camps. And then we had a wonderful team of staff. We had 5 permanent project staff and a bunch of collaborators, and a team of field assistants each summer that were hired to come in for the field season. And then this was one of 9 projects funded simultaneously by the Pollinator Initiative, each looking at kind of something bad that was happening to pollinators. So we did the urbanization, but there were pesticides and habitat loss and agricultural intensification and so on. And it was a really inspiring kind of body of, people and research that we got caught up in here, which was just, it was a lot of fun. So starting with this first question, where is the pollinator biodiversity in the UK? Which of those 3 habitats? So to do this, what we did was to sample plants and pollinators in 12 cities, 12 farms, and 12 nature reserves across the whole of the UK. At each of our 36 field sites, our plots were 1 kilometre squared. We had a lot of GIS, kind of global informatics system, help with sorting out these field sites. Each site is 1 kilometre square, and then within each site, we sample the different habitats according to their abundance, okay? And we did that for a whole field season. And so this is what our coverage looked like. We've got the Scottish team at Edinburgh, the Northern team, the Eastern team, and the Western team. And each of us had 3 focal cities, and each city was a cluster of a triplet. There was a city, a farm, and a nature reserve in a triplet, in a block design. And so overall, we have 36 fill sites across the country. And this is what it looks like. This is the Reading team. I can tell that from the people, not the road verge. But they're sampling a road verge in Reading, one of the many habitats we were looking at. We've got here the Yorkshire team in a nature reserve, and then back to the Reading team in a oilseed rape field by the look of it. So lots and lots of fieldwork sampling all these habitats, doing nothing very fancy, going out with insect nets and your data sheets and catching and collecting insects. And the end of that result, this is Kath Baldock, who was the lead postdoc, who collected about 7,500 insects in the field from the 36 field sites. Okay, so all go out to the taxonomists, all have Latin binomials put on them. And what we found, just briefly these results here, is there was no difference in abundance amongst farmland, nature reserve and urban habitats. So that was pretty, there's no difference. We found significantly more bee species in the cities than the surrounding farmland. And there's kind of two interpretations there. Either cities could be just wonderful habitats, that's the positive interpretation, or farmland is just so badly degraded that there's not much left living there. But overall, there's more bee species in the cities. But we did find rare species in all three habitats. So this work is published in, this is a Procroysop paper. So looking at now moving into a kind of a focused look at those four cities, Bristol, Reading, Leeds and Edinburgh. Where are the hotspots of pollinator biodiversity in cities? So this is, that's a bit of Bristol, and those are all the habitats that make up a city in most northern European places. You've got gardens, parks, road verges, cemeteries, urban nature reserves, allotments, which are community gardens. You've then got this other green space category, which is all those little bits and pieces of green space, which is normally mown, but owned by lots and lots of different people. We've got man-made surfaces, all the concretey stuff. We've got pavements. Pavements have good weeds, actually. Good things grow in pavements. And then we've got railways, roads, and water. And those are your habitats in a city. That makes up about 99% of the city. And we sampled everything except for 3 of them. We didn't sample water. There's no aquatic pollinators. There's some aquatic larvae, but not adult pollinators. Roads are too dangerous. We don't sample roads, but we do the road verges. And then railways, we couldn't get permission to sample. So there is good vegetation along the edge of railways, but you just cannot get permission to sample it. And it's a microscopic proportion of each city. So we had to leave the railways. But everything else we sampled. And we did this like this. So this is it's just a very systematic sampling program. Each city was divided into 10 regions using the census data. So it's actually stratified by income as well. And in each of those 10 regions, we had our 9 habitat types. So overall, you got 10 times 9, you got 90 field sites per city. Ooh, interesting. Oh no, back again, right, just, little blip, somebody blinked somewhere. So, where was I? 90 field sites per city. So, summarising, still there, summarising the sampling effort, we did 4,500 transect walks, collected about 8,000 visitors, 390 species of pollinator, 650 odd plant species, and about 500,000 floral units counted. I will just continue. It's just going to flick on and off. We'll just ignore it. It's just Microsoft or someone playing silly games. So, so the total data set is 360 local plant pollinator networks, a bit like that little Ecology letters one. Well, there's 360 of them in this data set, 90 for each of those 4 cities. So that's what our data set looks like. And these are the kind of overview of the results. There's an effect of land use on pollinator species richness and abundance. So we've got the mean richness per unit area at the side. And you can see that allotments and gardens are very good. Cemeteries and nature reserves, pretty good. And then by the time you get to the man-made surfaces, there's not much out there. So that this was kind of as expected with the exception of the allotments. Allotments, these community gardens, are normally thought of as kind of agricultural habitats, not very interesting, but they turn out to be superb for pollinators. But if you want to know, go back to that question about where the pollinators are, you have to know about how common these habitats are. So you've got your numbers of pollinator per unit area, and you need to know how much area you've got. So if you look at what a city is made of, it's actually, this was a bit surprising actually. So we've got the proportion by area of all of these different habitats here. So you've got Bristol in red, Edinburgh in blue, and so on. And they're remarkably homogeneous. So most British cities are a quarter to a third gardens. And they have a tiny amount of allotments. It's less than 1% of allotments. Remember that, because we'll come back to that. And then this other green space, which is this kind of mixture of all sorts of bits of green space outside social housing and all sorts of little bits and pieces, is actually a really large area again. And then there's, parks are quite small actually, cemeteries really small, but that's what British cities look like. So what you need to do is multiply the area of each habitat in each city by the number of pollinators, and that tells you where the pollinators are in British cities, and by extension, most Northern European cities. So we've swapped the y-axis here for the number of visitors per habitat, and let me just take you through this. And then we've got the habitat type along the bottom. So start with gardens. So it's kind of conceptually quite straightforward. You've got a large area of gardens. Remember, they're a quarter to a third of each city, and they're packed full of pollinators. So most pollinators are in gardens in British cities. If you look at your allotments, they actually had, again, very high numbers of pollinators, but it's a tiny area, 1% of the city. So actually it's city-wide as a system. We're looking at this as a city system. The pollinators, there are not many of them coming from allotments. But what you could do with allotments is you could actually have a lot more allotments. You can't have a lot more gardens because there isn't space for more gardens, but there's definitely space to double or triple the amount of allotments in cities. Parks are actually not great. There's potential for improvement as they could be really a lot better. Okay, they're kind of okay for pollinators. They're a medium sort of area. And then the real opportunity is this other green space. This is both, it's a really large area and it's really bad for pollinators. If you just made it a little bit better for pollinators, you could actually make a difference in a city. So these are the sorts of things we're able to tell our conservation biologists, our collaborators on the nature reserve front. So moving on to the last question, how do we improve pollinator biodiversity and abundance within urban habitats? Well, there's two things you can do in this sort of scenario. You can either increase the quantity of the good habitats, so more of the good stuff, or you increase the quality of the lower habitats. Ideally, you do both, but those are the two kind of extremes of what you can do. And what we did was to use a modelling approach with our physicist collaborators, because these are models way more complicated than certainly field ecologists like me can do. We used a Bayesian framework for these models, and I'm going to tell you about the first one, increasing the quantity of good habitats. We did both, but I'll tell you about the first one. And our response variable is, rather than actually using species richness and abundance, which are the kind of normal sort of metrics, we're going to use network robustness as our metric, as our response variable. So just a quick revision session of what robustness is. It's actually a system or a community's response to species loss. And if you imagine a network of interactions, if you start removing species, how many linked extinctions are there? If there aren't many, that community is robust. If you start getting extinction cascades, it's not. So that's what robustness measures, that ability to withstand species loss. And the reason it's important is that a high level of community robustness to species loss is increasingly recognized as an important goal in restoration ecology, because robust communities are more able to withstand perturbations. And if you look at everything that's happening out there with climate change and all the city-specific issues there are, you need robust communities in your urban ecology, urban pollinator community. And you simply measure robustness as a network statistic, and it has a value between 0 and 1. Now, I'm not going to go into the minutiae of how the models are done, but the actual conceptual framework is reasonably straightforward, actually, and it's quite an interesting way to work. So this is our city with the 9 habitats that we measured. For each of those 9 habitats, we now have a network. Okay, and there's 10 of those networks, so our city as a whole has got these. If you visualize that with 90 little networks all over it. What we start doing in silico, what our collaborator Philip Stanienko does, is he starts killing off species according to a whole series of biologically informed rules. So when you start removing the plants, you can sometimes get linked extinctions. At the same time, insects are highly mobile, okay? Pollinators can fly. So we also have dispersal between our different habitats and our different networks going on. And as broad rules of thumb, bigger insects disperse further than smaller insects. And there's really good regression relationships for that. So all of our insects got measured. So we've got some index of some measure of their disturbance ability. And what you're doing is you're running all of this and asking, if you increase the quantity of all our habitats one by one, which habitats make the best difference? If we had more of them, what would make the most difference to the robustness of the plant pollinator communities? And we did this, of course, we've got 4 cities worth of data to do this in, so we've got replication. And we're increasing the quantity of the good habitats. And this is what we found. This was a surprise, actually. The area of the allotments, increasing the area of the allotments, these tiny little species-rich habitats, made the biggest difference to these city-wide pollinator robustness networks, pollinator robustness variable in 3 of the cities, and the second biggest difference in the remaining city. And that's kind of an incredibly useful result, because actually we could increase the area of allotments, because it's only it's less than 1% in each city. So even in a crowded place like Bristol, we could have more allotments. And the nice thing is that they're not only good for pollinators, allotments. There's all sorts of data showing that people are happier and more kind of balanced in their views, and their stress hormones are lower if they have an allotment. So allotments are really good for people too, and they're also really good for sustainability and things. So you've got this win-win situation. So there's lots more results in this, but I'll move on to the next project now. So, sticking with lots of pollinators, we're looking at conservation at the landscape level. I'm starting with a little bit of background and the questions. So, conservation is increasingly done at the landscape level, but actually a lot of these interaction networks, including most of my own, are habitat specific. So they're kind of urban networks, farmland networks, heathland networks. They don't look at the whole landscape. And what we wanted to ask was, if you have a mosaic of habitats in a landscape, which you generally do, how does that affect community structure, so the architecture of the networks, but also community function? How well does that landscape actually work ecologically? And the kind of, in a nutshell, the question is, are landscape communities simply the sum of their habitat parts, or are there emergent properties such as increased stability or functioning that come with having a mixture of landscapes there? And that's what we set out to test on this project. So visually, what we're doing, if you imagine this as a very simple sort of cartoon of landscapes and habitats within them, you've got a single landscape there, 3 habitats. Does it look like that, with little food webs associated separately with each habitat? Or is it more like that? Because there's a difference in how you want to manage those landscapes according to which of those 2 it looks like. So we were asking which of those landscapes we were working in the UK, what do they look like? And this is how we did it. So we're actually using habitats that are there naturally. We're not using little plots of things. We're using the real world, which has both advantages and disadvantages, but mostly advantages. So here we've got a matrix of farmland, and it's got little patches of 3 different habitats in it. It's got grassland, heathland, and woodland. And our treatments, each field site is exactly the same size and size. All our field sites are 9 hectares in size, and then we've got these 3 treatments. We made up kind of names for these treatments. We have our monads, which is a single habitat, which is 9 hectares in size. And we have our dyads, which are 2 habitats, but they're half as big, so the total plot is 9 hectares, but our dyads are 4.5 hectares each. And then we have our triads. So our triad is grassland, heathland, and woodland, and they're 3 hectares in size, but our total plot is still the same size, okay? So there's no confounding effective area. So that's those were our kind of comparisons. We have 10 replicates of each of those. So again, some really hardcore GIS work to find these in the landscape and then ground truth them, because GIS isn't always right, actually. And so this is where we were working in the southwest of the UK. Our field sites are scattered around there. There's quite a few coastal ones because we're working on quite a few coastal habitats. And we end up with 30 independent field sites with these 3 treatments. And the one bit I've not told you about yet is there's no confounding effect of habitat identity. Because if we stuck with just the 3 habitats, Well, monads and dyads will be really variable because they're different pairwise combinations or different individual habitats, and our triads will be the same because they're the same 3 every time. So to avoid that, we picked our treatments from a pool of 6 habitats, so our triads varied a lot as well. So we're working in woodland, salt marsh, sand dune, heathland, meadow, and scrub. Okay, those are our field sites, or our field habitats. And then within each of our monads, dyads, and triads, we sampled 4 types of communities and their interactions. So we always have plant pollinators in there. They're very straightforward to collect the data for, and they're very important. And about 50% of our data comes from plant pollinator communities. But we also collected data on 3 guilds of herbivores. So we had caterpillars, all the butterfly and moth and external beetles and things like that, and they're parasitoids as well. So externally feeding larvae and their parasitoids and the plants they eat on. Then we had leaf miners and their plants and their parasitoids. And then we also had the seed feeders and their parasitoids. And actually, as in the Norwood project, these are joined up. Because if you think about it, your larval pollinators, if you're a butterfly or a moth, you're going to get parasitoids at that point in life. And you don't have seeds unless you have pollinators there anyway. So it's all one kind of joined up network of networks. So we're working on those 6 habitats in 30 field sites with these 4 types of community. And so what we're doing, this is a bit like a military operation running these sorts of projects. So we're collecting species and interactions at each of the 30 field sites. We're collecting data on plants, on pollinators, on herbivores, and on parasitoids, and on all of their interactions with each other. And then we're using that data to calculate network level metrics such as robustness. Robustness is one of my favorites because it's ecologically meaningful, actually, is why I like it. Now, actually, anybody here that's done field work knows there's days when it's just the absolute best job in the world, being a field ecologist. You are in beautiful places, doing interesting things with wonderful other people, and it's just absolutely, it's lovely. It's what we call the life of Riley, okay? It can be fantastic. Equally, if you're a field biologist, you know there are days when it's actually not much fun at all. We have to work in the British summer, okay? Today is a really, in Britain, I know the weather forecast is terrible today, it's cold and wet. I mean, really, trying to collect insects when it's raining, when it's cold, and I think all of us can sympathise with that poor person in the bottom photograph, and you just can't bear to count another caterpillar or plant or whatever. So it is a mixed blessing being a field ecologist. But the good days outnumber the bad days, okay, always, always, always. So that's the field work. This is what the raw data look like. So these are what are called multiplex networks. You stack your different habitats, a bit like a pile of pancakes, basically. The links between the layers occur in these networks when species are shared between the habitats. And these are some of our real data. So we've got data from a monad at the top, a dyad, a woodland and a salt marsh in the middle, and then a triad, scrub, heathland and grassland down the bottom. And we've got 10 reps of each of those sorts of data sets. Now, looking at the results, starting off with the simple metrics, we're looking at things like plant species richness, floral abundance, insect species abundance, species evenness, and interaction evenness. And broadly, and as you'd expect, actually, these are higher when there's more habitats. So if you've got more habitat specialists and you've got more habitats, you're going to have more species. And the significant differences are mostly between the monads and the triads. The dyads are somewhere there in the middle. So that's the kind of descriptive side of what's going on in those 30, whatever it is, networks. When you start moving on to robustness, things get a bit more subtle and a bit more interesting. So remember, robustness is that measure of how communities respond to species loss. Do they, not much changes, or do you get extinction cascades? And what you've got here is robustness on the y-axis going from 0 to 1. And you've got our monads, our dyads, and our triads, and the small print is just our local names for them. And then what you can see is that there's no difference in mean robustness between our monads, dyads, and triads. Robustness is fairly high, but no difference in mean robustness. But there is a significant difference in the variability of that robustness, and our monads are significantly more variable than the triads. And that's important. As ecologists, we sometimes get a bit fixated on measures of central tendency on means and modes and medians and things. And actually variability is really important. And in this case, it's particularly important because the higher variability in these single landscape habitats in our monads means that extremes of robustness are actually just more likely to happen. They're more common. And so those communities at those individual sites are more frequently at risk of extinction cascades, at least according to our models. Right, so that's community architecture, the structure of the nodes and the links and the network metrics. What really counts is how well, I mean, that's important, but community function is really, really important. So here we're going to come back and focus on the pollinators and the ecosystem function of pollination. So what we did here was we ran a big field experiment, probably the most ambitious field experiment I've ever run, looking at differences in function between our monads and our triads. So we looked at the two extremes. We used strawberry plants as a bioassay for pollination, and we'd done this on 2 other projects previously. And it's just one of nature's happy accidents that strawberries are really good for measuring pollination with. And that's because next time you eat strawberries, you get your punnet from the supermarket, you'll find at the bottom, there's nearly always a couple of these kind of non-symmetrical strawberries. At the top, they put the beautiful round, plump, symmetrical strawberries. And then lower down, they always hide a couple of these, of the distorted ones. And what's happened is, in the plump, symmetrical strawberry, every single ovule has been pollinated by pollinators, okay? They have multiple seeds, multiple ovules, and if they're all pollinated, the red stuff, which is what we like to eat, swells exactly the same on each seed. If they're not evenly pollinated, so there's still some ovules that are unpollinated, you get this kind of asymmetry in the fruit. And commercial strawberry farmers grade their strawberries. So the round ones go to Wimbledon and the posh supermarkets, and the class 1 strawberries, and the less posh strawberries go to make jam. Okay, so this will taste the same. But it gives us a very convenient way of measuring pollination. So we've got grade 1 and grade 2 strawberries. So in this experiment, and this is a Herculean experiment, we put 16 strawberry plants in our monads and in our triads, okay? So it's 4 buckets of soil and strawberries going to each plot. They have to be carried through woodlands and over fields and left in the plot, protected from cows and so on. And they're left for 2 weeks. And we did this, we've got 10 replicates of each habitat type, remember? So we've got 20 triads, 20 monads, left them out there for 2 weeks, then carried them all the way back through the woods and the fields, put them in the car, brought them back to Bristol, and they were kept in a greenhouse until the fruit ripened, okay, so free of pollinators from then on. And this is what we found. So we've got data on fruit weight and fruit symmetry. Fruit weight, there's no significant difference. There's a small trend, a slightly higher one in the triads, but it's not significantly different. The fruit quality, though, the pollination quality, which is what we're measuring, is significantly different. So there's a higher proportion of class 1 strawberries in our triads. They're having a better pollination success when everything else is the same. It's just the number of habitats around our buckets of strawberries. So significant difference in pollination function in triads compared to monads. And what this sort of thing means practically is, as practitioners, you need to consider the bonus effects of having multiple habitats. So in Britain, we have this project called the Living Landscape Project. These are big projects, but they focus on particular habitats. So around where I live in Bristol, it's calcareous grassland that people actually focus on. So lots of work on calcareous grassland. Go to the States, there's loads of prairie restoration projects, but we really do need to consider the bonus effects of having multiple habitats, because there's some real advantages. And if you think of pollinators, if you've got a woodland, a grassland, and a heathland, the woodland flowers in early spring, the grassland in kind of early summer, and the heathland in midsummer. So you've got this continuity of resources for pollinators. And the same applies to lots of other organisms. So this work is currently in Nature in Press, and this is just, you know, the team of people involved in making this work was, there's some amazing people here. We've got field biologists, we've got modelers, we've got the technicians. It was a really big, exciting bit of work that we just got coming out this summer. It's a big relief to get this one off my desk. Right, so the last project I want to tell you about is actually one that's in a different stage. It's been running for the last 3 years. We've just had enough money to collect the data, no money to process anything. And we're always thinking we were going to run out of money, but we just got proper funding to run it for another 4 years. So it's a project that we're really bonded with already, and we've now got the money to take forward. So the 3 questions here, and pesticides are a new topic for me. It's always fun every now and again to start to do something really new and different. We're interested in the effect of a pesticide ban on pollinator populations and pollinator communities. So we're looking at individual species and at the whole community. And then probably the most exciting and also the riskiest and most ambitious bit is to ask if there's cascading impacts on the wider ecological community if you restore pollinators. So looking specifically at the things that eat pollinators, so things like generalist predators such as bats and specialist predators such as the canopid parasitoids that feed on bumblebees. So our field sites we're working on an archipelago of islands just off the French coast. So it's Guernsey, Jersey, Alderney and Sark, which are just beautiful places to work. And I can get there in 40 minutes from Bristol. And yeah, I used to work in places like Hawaii, and it's just so nice to have a really local archipelago. So based on those islands, and the backdrop to this is a really inspiring story. So back in 2017, a group of volunteers with science backgrounds, ecology, wildlife backgrounds, and business backgrounds formed something called the Pollinator Project. These were people that were really keen on conserving pollinators. And their aim was to promote and protect pollinators in Guernsey. And a key part of their proposal is that Guernsey should become pesticide-free. Okay, they wanted to do that for a variety of environmental, and also kind of for tourism. It makes the island look really green. But it provided a unique opportunity to study the impact of pesticide bans on the scale of an entire island. Because up until this point, the way we've looked at pesticides is you can poison pollinators in petri dishes in the lab, you know, one by one, and see when they die. That's been used by the pesticide companies for a long time. And there's a handful of studies done at the scale of a field, you know, a field of oilseed rape. But there's really not very many of those sorts of studies, and they're still just done at the field scale. This became a chance, a one-off chance, to look at it at the effect of a whole system, a whole island. Guernsey is about 8 by 12 kilometers, I think. So it's, you know, it's a decent chunk of field site. So what this small group of really dedicated people did was in 2019, there was the first stakeholder consultation. So you have to talk to all the people you want to get on board. Talking is really, really, really important. In 2020, they had the first pesticide amnesty, so a bit like a firearms amnesty. They got all the people to bring the things at the back of their sheds and hand them in to be safely disposed of. And actually, DDT came out the back of someone's shed, and that was banned in the 1960s. So people don't tidy out the backs of their sheds very often. 2021, the National Trust, which is either the largest or the second largest landowner, stopped using pesticides. And this is all due to this small bunch of people. 2022, glyphosate, Roundup, was banned on public land. So all the schools, hospitals, etc., etc. 2023, full auditing of pesticide use came in by professional users. So it's just killing them by paperwork, basically. If you want to use a pesticide, you've got to fill out 50 forms first. So it's about reducing pesticide use. In 2024, domestic pesticide use is going to be phased out. Bizarrely, they sell pesticides in supermarkets on the Channel Isles. This is really weird, but they do. It's kind of not quite next to the bread, but you know, but it's not done anymore. 2 of the supermarkets have stopped doing that. 2 of the supermarket brands. And then finally, pesticide sales will be banned and pesticide-free status achieved. And then just to use one of my favorite quotes, never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it's the only thing that ever has. And so this is Margaret Mead, who's an anthropologist. So you guys, everybody, all of us can be part of this change. So what we're doing, and this is now going to be an 8-year project. We've got 4 years data in the freezers, and we've got funding to collect another 4 years post-ban, and actually pay for the processing of the first 4 years as well. So on our islands, we have 16 malaise traps in what's called a BACI design, a before-after controlled impact design, which is a way of it's an elegant statistical way of getting around a small sample size. because we've only got one experiment, Guernsey, and those will run from 2021 to 2028. And then we're also looking at the bumblebees. If you count adult bumblebees, that's not your reproductive, that's not the unit you need. You need to know the number of queens, which means to know the number of nests. And to do that, you use a microsatellite genotyping approach. And we're collecting a single foot from Bombus terrestris on all the islands in a very systematic way. So by the end of the field season, there's a lot of 5-footed bumblebees on the Channel Isles if you go out there. I'm afraid that's us. But they get to fly and live another day. So there's the bumblebees. And just to give you an idea of what this data can do, so the only sorting we've done so far of the data is we've done a rough sort of the first 2 years of Malaise trap data. And we pulled out, I had a team of students, because Malaise trap data is really complicated stuff. There's a lot of insects in Malaise trap. And we pulled out one of the commonest hoverflies, Episurfus balteatus, and did a power analysis on it. And we've actually got an 84% chance of detecting a 10% increase. So if the pesticide ban leads to just a 10% increase, we've got the statistical power to pick that out. And a 98% chance of detecting a 15% increase, which actually I find quite reassuring. And of course, we've got 8 years data on lots of different species, a handful of which are really common, like Balteatus. So the community level stuff, all we've done so far is do a kind of a quick ordination on the first 2 years data. And rather nicely, there's no difference in the communities right now. So these are the kind of community fingerprints, basically. And Guernsey is exactly the same as the others. And what we might expect to happen, though we'll use more sophisticated approaches, is that Guernsey will drift off to one side as its community changes due to the pesticide ban. But pre-ban, there's no difference. And then the final thing we're going to be doing is testing if there's cascading impacts of the ban at the wider food web level. So we're doing 2 things here. We've got bat detectors next to all of our Malaise trap sites. We're working with some 2 bat ecologists. And then also going back to my parasitoid days, pollinators have really good parasitoids. So bumblebees have canopied flies, hoverflies have diplozontine braconids, and your leps have ichneumons and braconids and a variety of others. So in our Malaise traps, we have lots and lots of parasitoids. So we can look to see if the parasitoids also recover. So this is, there's lots and lots of people involved in this. So universities, wildlife trusts, pollinator projects, states of Guernsey, all sorts of people. Right, there'll be a talk on that in 3 years' time when we've got the data. How to bring more pollinators into your lives? Okay, so what can you do in your back garden or your balcony or your doorstep, whatever you've got to kind of, whatever space you've got, you can use it. So this is what I do. This is my bit of my back garden. So we don't have a lawn at the back. We have a wildlife meadow. So it's actually far easier to keep. You only have to mow it a couple of times a year. It's far prettier, much nicer to look at, and it's really good for pollinators. We have nests for solitary bees. You can hear these as you come towards it. The buzz of the bees is so loud. Careful use of plants, so non-native plants are also really, really important to pollinators. So things with really long seasons and good nectar supplies, like this biannual perennial wallflower. And then I'm still rearing caterpillars just for the fun of it. So on my windowsill over the winter were 2 chrysalises from 2 elephant hawk moths. One of them, the weekend before last, turned into the most gorgeous adult, elephant hawk moth. And the other turned into the biggest parasitoid I've ever seen, which as a parasitoid person, I'm every bit as pleased to see. They are very welcome as well. So there's lots you can do on your patch. And even if you just have a doorstep, put some herbs on there. You can put them on your pizza on a Friday evening. And herb flowers are really good for pollinators. So you can whatever land you've got, put pollinator-friendly things on it. And then finally, if ever you need to impress your kids, your friends, or in my case, I'm working with a film crew next week from Gardener's World or something, impress them how kind of clever this relationship between plants and pollinators is. A really good little trick you can do using ingredients from your kitchen cupboard is to work with foxglove, Digitalis purpurea. It's a big plant that's in flower in Britain right now, common plant, wayside native plant. And if you stand next to a patch of foxgloves, next time you see a foxglove, what you will see is any bees will do something very predictable. They will fly to the bottom of the column of flowers. They will then go up, and they'll then fly to the next plant. Okay, and they don't all do it. A new bumblebee has to learn to do this, but most bumblebees will do this on any of these tall column-like flowers. And the reason they're doing that is there's more nectar at the bottom, in the bottom flowers, okay? The flower is putting more nectar into its lower flowers. And you start thinking, well, why do that? You want all your flowers to be visited. But the reason they do that is at the bottom of the column, the female flowers are receptive, so the stigmas want to be pollinated. Whereas at the top, the males are releasing their pollen. Okay, each flower is dioecious, they produce both, but they are receptive at different times. So what the plant is doing is training pollinators, and bees learn really quickly that there's more resources at the bottom, and so getting pollinators to do this increases outcrossing, because you're bringing your pollen into your receptive female flowers, and reducing inbreeding. And you can show this to your kids, your friends, and your film crews as follows. So try this at home. You need to buy what we call in Britain a French stick, so a baguette. And they come in British supermarkets, they come in these plastic bags, which are kind of permeable. You eat the bread for lunch, keep the bag, okay? Put the bag over the foxglove for 24 hours, okay, to keep the pollinators out, because bumblebees love foxgloves. Do that, then go back to your kitchen cupboard, get your coffee filter paper, and cut it into little wicks, little triangles. When you've done that, you can use that to suck the nectar out of each and every flower. It doesn't go red, but you can see the wet, okay, it's really clear, you can draw a line. And so I did one last weekend, and this is what you get. So this is the bottom of the foxglove down here, top of the foxglove up there, and the black line is how much nectar's in it. So at the bottom, the nectar goes right to the top of the wick. And if you go to the top of the kind of foxglove flowers, which is higher up the stem, there's barely any. So it's a slightly noisy, you know, there's the odd little glitch, but if you do lots of these, it's a really clear pattern. And if you look inside your flowers, you'll see at the bottom, the female stigmas are open and receptive, and at the top, the pollen sacs are starting to dehisce. So your take-home message for your kids, your friends, and your film crews is plants don't just sit there looking pretty. Okay, they train pollinators to get what they want. It's a really clever system. So, to end, brief conclusions. What we've done, we've looked at why pollinators are so important. We've looked at this community ecology approach for studying pollinators. And I've told you about 3 projects. One that's mostly finished, one that's almost finished coming out this summer, and one that's kind of really just starting off. And I've told you some top tips on what to do to get more pollinators into your everyday lives, because I can guarantee there is nothing nicer than being sat there with a cup of tea or a glass of chilled wine, listening to the bees buzzing in the flowers. It'll make you feel better about everything. And on that note, I'll end, actually. So thank you.
So do you have questions?
I'm just going to sit down.
Well, thank you very much. It was a wonderful talk, and I'm definitely going to try this with my children and lots of foxgloves. Just on the first part, do you have any idea why there's so you have lots of pollinators, lots of diversity in these gardens and the allotments? Do you know why there's such a difference with the parks and the other green spaces? Is it because of heterogeneity, flower abundance, or?
Speak it on my microphone, actually. Is this microphone plugged in? Actually,
I can hear you well.
Can you hear me well?
Yeah.
Right. A lot of it's to do with nature. So allotments are very, very variable. There's a really good mix of flowers. There's a really good mix of nesting styles. You know, bumblebees love nesting under sheds. You've got a lot of everything in allotments. By the time you get to parks, those British parks are mown grass. Okay, it's to do with management. We could do so much better. And actually, Bristol and Bath next to you, they let large areas of wildlife meadow now. So it's a question of you need more resources, basically. You get more flowers and more pollinators. You need food and you need somewhere to nest. And a lot was done by both of those in abundance, and many of us are habitats don't. And the reason why that other green space has such potential it's always known. It's like this high. You know, it's not even daisies on most of it. And what's ridiculous is it's expensive mowing. So if you just mowed less, and the other modelling we did was actually modelling the effect of mowing parts and other green space less, you also get an increase in robustness. So it's to do with the amount of resources, the amount of nesting sites. And both of those are fixable, is the kind of encouraging take message. We could do better. And it's why it's really important to talk to people that do the work that actually can make the difference, because they want more pollinators too. So certainly in Bristol, Bristol is now hoping to become the pollination capital of Britain, actually. And that's the local council writing that. We've got them so fired up and keen on pollinators that it does work. People listen and want to help.
Okay, thank you very much. I'm very impressed by this move on the island of Bristol. I think it's also the possibility that you have an island makes it easier to really to get a complete ban. And I just want to recall that in France, since 50 years now, we have in many of the municipalities, the program was called Zero Phyto, so zero phytosanitary products, and the municipalities were completely banning it, and this has been really successful. But on the mainland, of course, you have a situation that the green spaces managed by the municipalities, they don't use pesticides anymore, but in some cases they are able to convince the private owners, in other cases not, and you have besides the agricultural landscape where it's still continuing. So finally, from this pesticide study, would you also be interested to do some sort of landscape study where you compare areas where you have complete ban as on this island compared to other areas where you have the banned areas coexisting with areas where the pesticides are still being used?
Yeah, you should do it. It would be hard. The advantage of an island actually is a because insects migrate. So the genetics work will show that insects migrating from France to England, that certainly be going to the Channel Islands. Interestingly, things like the tree bumblebee, almost ignore them, isn't on the Channel Islands for reasons I don't understand. You can do it, but it'd be hard. You want large areas where you've got your treatments. And the thing that makes it particularly challenging is, of course, insects come and go from those areas. Pollinators are mobile. So if you focus on your central place foragers, which aren't as mobile, let's say your big hoverflies, that would be your best bet. Just focus on small central place foragers like the solitary bees, because actually, 25 years ago, most pollination researchers were bees. Then the bumblebees were kind of invented, and people started working on bumblebees. But the thing we really don't know about are the solitary bees. And that, with grass coding, they're not even that hard to identify now. So my advice is to work on a central place forager that isn't just drifting across the landscape, like say the hoverflies do. And yeah, potentially, if you know where they're definitely not being used, and you know where they are definitely being used. And actually, that's sometimes quite complicated because people say they're not using them, that's what you do. And then also, you kind of, people don't remember what the it's complicated getting data out of people on pesticides. But you could do that. If you get enough, I would just have a lot of records would be my top tip, because the data will be noisy. It could be done. So, but the island system is just a gift from heaven. It's just, as I say, it's just wonderful. It's rather than keep it some really dedicated people, and to be able to work with them is pretty special. But try it on the mainland. If you've got one of the nice things, we haven't done it yet, but one of the collaborators on the project is the water board, the Guernsey Water Board. Because they don't like pesticide because they have a reservoir system, so they wash off into the reservoirs, which nobody likes. And what's interesting, Guernsey, one of the downsides of it, even though it's mostly perfect, is that most pesticide use is probably gardeners. Okay, the farms, there's 12 farms on Guernsey, and they're all dairy farms. And so they wouldn't use a bit of stuff, not that much. But gardeners, you get spikes of pesticides on the water after a bank holiday. It's like drug use in Bristol, the spikes of drugs which on Monday morning for all those parties. you get spikes of pesticides in the water, so it's a kind of an urban problem, but it is a really good experimental system. But try it, I'd love to see it.
We have a question about the conservation of the landscape level. How do you determine the right spatial scale at which heterogeneity matters and we want to maintain diversity of species?
Good question. I don't know. I mean, we know at the 9 hectare level, which is actually a very small landscape, but actually when you're trying to replicate it 30 times, you have to do something that's practical. My guess is that at that level we are picking up the effect, then that's kind of, we should be thinking about this as a small sort of scale as well as a really big scale. Because 9 hectares, although it's big, it's not a landscape per se, but it's a very useful kind of mesocosm for landscape. It's the biggest mesocosm ever, actually, 9 hectares. So I think we don't, to do that, would take a different approach to gathering data, gathering that sort of data at different spatial scales, and then you can start to see where it kicks in. As the broad, I mean, there's always there's a saying in English that excellence is the enemy of good. So trying to do it perfectly is you can't never do that as an ecologist. You have to, there's always some data you can't get. We know that at the scale of 9 hectares it makes a difference. My advice to nature reserve owners is, one, if you've got a landscape front horse, have some more than one habitat in it. Look at the adjacencies. So if you're buying a nature reserve, it's not just obviously your nature, look at what's around it. So they're very practical take-home messages. The bottom line is we don't know the exact spatial scale, but another day, another dollar, you could actually sample at different spatial scales and start to work out where it is. But use 9 hectares as your minimum would be a basic place to start.
Thank you very much for your talk. I was wondering, in the urban pollination project, you show that clearly we can do some land use changes in cities. Has been in these different cities any change that has been really done? Because you can provide, for example, recommendations. Let's cut the grass often, for example, but changing land use to give green land to buildings, that's more difficult, I guess. So, has there been any kind of change?
There's not a systematic concerted change, but what there is, so Bristol City Council has put £500,000 into prizes for Pollinator projects. So there's lots of little projects all doing their separate thing. And my slight worry is that there's lots of little differences, and actually doing something really big once might make a difference. We don't know, but what we will be able to do is, because that data that was collected in, I think it was 2012 and 13 or something, that is your baseline level. So if we can say in another 5 years or so repeat that standard, because we've got all our Pollinator, what the field sites were, and do that again a bit like the API design, where you have Bristol, which has done quite a lot, and then go to 4 cities where. But the problem is that everybody's got pollinators. There aren't many cities which aren't doing anything. So it's a tricky one, but I would really like to actually use that baseline data as to do something else with. One of the nice things that came out of that project was Graham Stone, who was on our team. He got all of the specimens from all of the projects. I've got those catalogued at the Natural History Museum. So the whole lot, the specimens on hins with all the genetic information in are there as a resource for future projects. So it could be done, but the answer is a direction of travel, and there's a lot happening, but it's not enormously done. So I'm on the prize getting university, so I do my best. But yeah.
Thanks.
Yeah.
Thank you very much for your impressive talk. I'm very much impressed by the amount of data as well. And I would like to ask a question. You have described the interaction networks on a taxonomic basis. Have you tried anything about the trait interaction?
Yeah, we do. So it's only pollinators, it's very definite, right? So I put some in the landscape project we did some that. It does get you around some of the issues of, so I always think the gold standard is to try and get a Latin binomial on things. You know exactly what's there. That is just useful information. If you've got rare species, it's much easier to get conservation action. Even the rare species technically aren't culturally that important, but it's a useful lever. So as our bottom line, we always start with that taxonomic Latin binomial, but we will other things into traits, and some other projects have done that. And it does actually speed things up. If you put, you're setting your bees into tongue lens, use taxonomy, there's things you can do. There's like plants as well. So yes, we do, but we tend to, we're pretty big it might be a bit too detail obsessed, I sometimes think, but it is definitely for practical conservation purposes. It's incredibly useful because they've never got the resources to do that at taxonomic level kind of identification. So yeah, it's the way forward actually. There's a lot more on that. So the brief answer is yes.
Moving for the talk, I was interested about what you said about the results on farms, that you found less diversity in farming. I was wondering if it was because of animal treatments against parasites. Like, do you think it could be this? And because of that, I was wondering, regarding what you have done, if it's a good idea for cities to help, like to ask for farmers to take their animals and graze into the cities, you know, to...
Yeah, first question you asked, we don't know because there's lots of things that happen on farms that are bad for pollinators. So you have habitat loss, you have pesticides, you have intensification of variability. You know, the work we did on nectar, we quantified about nectar loss since the 1950s through to the early noughties, and a third of the nectar in the UK is gone from farmers. You know, that's from farmers and nature reserves, and that is due to intensification of agriculture, and that's a multitude of things. We don't know which particular bit, if any, or whether it's all of them collectively. It's probably a bit of everything. The insecticides, the animal type thing, what you've got is over thinking treating animals with the right thing is to do with other communities. So certainly for decomposing communities, that's really important. On Guernsey and in lots of other places, if you've got a dog or a cat, you're putting flea treatments, tick treatments on them. And vets are telling you to do that, just stop your dog getting ticks anyway. Those wash off into water, lots of dogs like hose water. So if you have a dog, only use it when you really got a problem. But actually, you can get little lice to take ticks off. And I've got a friend who I my ticks to even, I have strange friends. But you don't need to kind of use those things. So don't get into the water and they kill a problem. They're not pollinators, it's really not good, so I'm not keen on that. But on farms, it's just too complicated. So it's much about climate change, everything else. So we don't know what's the bottom line. As farming becomes, there's some quite interesting farming changes coming into Britain with the new environmental land management. So farm is 70% of the UK. So if we fix farms, we fix a lot of our biodiversity issues. And we should definitely be talking to farmers to do that. There you go.
Thank you for inspiring talk. And I was wondering for the farms, what type of farm did you sample? Did you compare some farms with high intensity measures or also agroecological measures or other type of farms to have more?
Yeah, what we didn't go for we went for what we call the moral standard farms, so they weren't organic farms. These were farms that were regionally matched. So you do different types of farming in the southwest around Edinburgh, Glasgow and things. So they were local, typical farms, and we deliberately didn't use, I've worked a lot on organic farms in the past, but we deliberately didn't use organic farms because they're not typical, sadly. So they're very standard sorts of farms, but I want to think back 12 years to remember exactly how we chose them, but we chose them very carefully, I remember that. So what you're actually looking for, what you guys are just coming back to me. So in our cities, our nature reserves and our farms, what we're actually asking is, do the GIS approaches. For a city, you look at all of the habitats in them. Okay, there's X% of gardens and Y% of allotments and things, and then in your 1 kilometre squared, you try and get those percentages as matched to the city as possible, and we'd have done the same with farms. So there were some quite complicated GIS approaches, but the pragmatic one we did to avoid organic farms.
Yeah, so someone mentioned the enormous amount of data indeed you have collected. So I've been doing ecology when I was a student, and I was kind of struck by the fact that mostly data from studies wasn't available. Has that changed? So is these data already available, or are there databases where you put this now? So this is an outsider question, but just to...
Yeah, that's a really good question. Actually, the culture has really changed. So the only pollinator data sets they should be available online. The landscape level one that's coming out, that data is all on. It's in the bottom of the paper. I've seen it. It's there. That is made available. Some other big projects I've not spoken about. Some of the core projects is we've got data for over 5 years. And actually, that's funded originally by something called the Memmot Foundation, and which is actually they get a bunch of funders from each country to work with 2 other nationalities and so on. So anyway, we do this project. But they are really, really big on data management. Now, actually, there's a lot by doing that. So all that data goes online. That goes on the NERC, EICD national database. So absolutely everybody, even if your experiment hasn't worked, whatever, make your data available, because there's huge amounts of data that have, they just disappear. They go into unpublished thesis. So we do try and do that really properly now. And we've definitely got better. Because when I started that 25 years ago, one of the things I would do when I was like one day, is go back through my old data sets and put those online, because we still have some of those. So, but certainly, in the last 10, 15 years, we've made it available. So, yeah, it is really important to do that. Medics, the genomic people, they always make their data completely available. That culture went in from the beginning. And this idea that someone's going to, steal your data, it doesn't happen. We've had an embargo on this, so our report data is embargoed until November, which is the complete end of funding, and our work is still the act review, and nobody will catch up on this in that space of time. It's taken us 2-3 years trying to price that data. So, and there's always this feeling that you're going to use it on a rainy day and something else, but most of the time you don't. So, 2-3 days until you finish, put the data online. Do the public spirited thing. It's important that it makes a difference.
Other questions?
Thank you for the presentation. I was wondering, because you said, I found pretty interesting how you end with this recommendation. How do you find the pollinators in the garden? But I don't know, I was wondering how about all the cascading effects that you were studying, and how do you also sensibilize this kind of groups of insects? So
good question, actually. So, we've got pollinators because it's easy to cut there. Yeah, you get a from parasitoid. Parasitoid is wonderful. Collecting the data takes a lot longer. You have to rear the parasitoids out individually in pots, and you want to look after them. So it's like zookeeping on a massive scale. You have 1,000 insects you're looking after. Pollinators are really quick. You just collect them in the field and identified. But if you're working on native plants, and so the Aurora project looks at all of the things on them, but if you've got native vegetation, if you've got natural vegetation, it will broadly be good for many different groups. So if you're a specialist herbivorous insects, for the parasitoids on this specialist herbivorous insects, you'll get things. So we've not quantified those in urban habitats, but I'll eat my hat if then, the sort of things that for pollinators will be generally good. But ideally, one of the things we've been trying to do over the years is break down some of those taxonomic barriers between different types of ecologists, because you've got the entomologists, the bird people, the mammal people, the marine people. There's still really big divides between terrestrial and marine people, and between freshwater and terrestrial people. And the few intriguing bits of the page out there that show there's actually really strong links between them. Nobody ever spots them because nobody works on both of them at once. So there's lots of scope for that. But broadly, I think in urban habitats, if you get naturalistic plantings in, those flowering plants have leaves and roots and stems, and they provide a habitat for lots of other things. But it's another day, another dollar to quantify all of them. I think people have done things on caribou, so generalist predators. But yeah, there's more to be done, for sure.
Other questions?
Maybe I have a question actually about domestic bees and the impact on white pollinators. Did you study this impact, competition for nectar, for example? Because I guess it can have a huge impact.
Yeah, that's really interesting. So I've got a PhD student who's working full-time on it at the moment. And if you look at the literature on honeybees, you'll find it's really variable. Some studies find really clear effects, and some studies find there's no effect. And it's really difficult to decide, well, do they, don't they have a bad effect on wild pollinators? What is probably happening is it depends where you measure. A lot of the time, there's a lot more nectar than pollinators. So if you imagine like Moorland, where the heather, the Kaluna is in flower, you know, there's buckets of nectar out there, and it's really not a limited resource. But early spring in those systems, it very probably is. So depending on when those studies take place, you either try competition or you don't. So what my student Maisie Brett is doing is she's measuring things over an entire year. She's doing that in 2 habitats. She's doing it in moorlands in Scotland, and she's doing it in another irrigation habitat, which is the Veinboss vegetation in South Africa. In Scottish communities, it's Apis mellifera, which is an alien, you know, it's not native to Scotland. That are kept there by beekeepers. And in South Africa, it's a native honeybee, it's Apis cerana, the native one, but it's maintained at very high numbers artificially. And she's managed to do an experiment, a false experiment in Scotland. She's got beekeepers to put out hives, large numbers of hives in particular places. And again, we've got a design that will allow us to unpick over an entire flowering season, from the first kind of very early springtime plants right through the end of the season. And she's trying to understanding properly both habitats, asking how much the wild pollinators need, and then putting the honeybees in there, and asking where the kind of pinch points are. Where will there be if there is competition? So it's a complicated one. There's quite a lot of work done on it, but it's a really mixed picture. But I think it's a mixed picture because people have been working on the wrong temporal scale. So watch this space. A couple of years we'll know.
Thank you. Please. Yeah, Jean, do you want to close the session? Okay, so maybe, so let's thank again Jane, and I have an announcement after that. Thank you all for being here. So now we'll have a buffet in the staff, really like 10 meters away. And then an announcement about the Oliver Conference and Lab. We still have the same meeting until early July. And then we'll have the Oliver Conference at the start of the session in September. And so Thomas and Memmot will give the Oliver Conference this year. So please come then in September. Thank you.
Thank you. So I couldn't see the screen coming and going, but it went. No. No, it wasn't worth fixing. Yeah. Hopefully internet or something. So, yeah. Right.
It's just searching for connection. It's not something we